welcome all. I'm Harold Trick, and I'm going to be just giving you an overview of the centralized wheat transformation facility here at K-State. And I guess what I'll do, I'll just go to my slides. Let me see if I can do this. Share. Okay, I think it's sharing now. Can you guys all see the slide?
Yes.
Okay. All right, that's good. Okay, so I've got it
Oh,
coming up.
Harold, it's like in presentation mode though.
Yep, that's what
Oh,
we
that's what
wanted,
you
right?
wanted? Okay.
Because yeah, so you can see the whole slide there.
No, we don't see the whole slide. We see all the
Yeah, we
upcoming
see
ones.
it like you see it, like as you're presenting with like the next.
Okay, hold on one second then. I just stopped the share.
Yep.
There we go.
There
That's
we
it.
go. Perfect.
Now you can see it. Okay. All right. So the centralized wheat transformation facility, I also have gene editing facility here at K-State. The reason why this was initialized with the scab community was we have a lot of specialized functionalities going into trying to create transgenic wheat or gene edited wheat. And we felt that all that redundancy would actually take away from um, the amount of money that everyone is able to utilize for their research projects. So that's why this was initiated. The idea would be for researchers associated with the SCAB initiative to send us their gene constructs. We'd actually engineer the wheat and then send back out the seeds to you all. So that was the sort of the underlying concept. And this has been going on now for probably 3 or 4 funding cycles. It's been several years now, and I think we've been having some pretty good success with this. But I wanted to go over sort of what we actually do for this. For trying to get onto the website itself, this is the U.S. Wheat and Barley Scab Initiative website. If you look where I have, let's see, do I have my tool here? That's my drawing tool. This is probably a drawing tool. Well, go back. Okay, the research tools, if you select that cursor and then draw down, you'll see the transformation labs. That's where you actually go on the website to actually see the site itself. The transformation labs, we've got 2 of them. We have one for barley transformation. This is Rong D at Rutgers University. And then on the bottom is mine for the wheat transformation. I do have a wheat submission form that I usually have the PIs fill out for this. And that's here what I have located on this slide here. This is what the submission form looks like. Essentially in this, you put in your name. If you have your U.S. Wheat and Barley grant number or ARS agreement, we'd like to have that up there so that way we can track what outputs we are getting from our grant to how we're impacting other grants. You also need to have an IBC number. If you don't know what that is, it's an Institutional Biosafety Committee number. That is from your institution that will let you know that you are allowed to actually do work with transgenic material. Okay. Again, with this, you'd put in your plasmid name, concentration, the amount that you want to give us, what cultivar you want that to be put in. So we have a number of cultivars. I'll go over those here in a little bit. But it's really up to the researcher as to what you want to put your plasmids in. We would like to also have information as to the gene or the phenotype below. Is it lethal or is it a knockout? This actually helps us when we actually do the transformations. If something goes wrong where we're not getting transgenics back for you guys, this may actually answer the question for us. It may be a mutation that's actually lethal to the plants. All the information you give us also is confidential. We will not release that to anyone else unless we have your permission. So you can rest assured that your information will be confidential in that situation. And so this is basically how we actually go about doing this. If you're unsure of this, you can always contact us ahead of time. You can use my email address. That's also on the website. And that way we can actually talk about your project a little bit to try to help out with what you want to actually accomplish with your research. Okay, so you would send us your plasmids, and that's what we would actually work with. With your plasmids, usually there's something called co-transformation. You can either have your gene of interest and your selectable marker on the same plasmid, or you can have them on separate plasmids. So sometimes people are unsure of actually how to actually combine those two together. We had the selectable marker, which is BAR or biolophos resistance, on a separate plasmid. And if you do not have your selectable marker on yours, we'll actually combine it with ours when we do the transformation itself. Okay. So for the transformation, what we go through is we do weak tissue culture. We start off with immature embryos. These are embryos that we grow in the growth chambers. These plants are about 60 days old or so, maybe about 50. Anyway, with these, the embryos that we capture are 10 to 14-day old embryos. So they're immature embryos. We will actually excise those out, put those on media. And then this is the whole process with this. We induce them to actually proliferate callus material. And that's the material that we actually genetically engineer. Then we move those on to selectable marker media where we actually start developing shoots from that material. We actually then go ahead and recover small plants from that material, and eventually we'll actually have plant recovery. So that's sort of the underlying as to how the tissue culture goes. For the transformation process, we use the gene gun. So this is our homemade version of the gene gun. It's a particle inflow gun. Can you see my cursor? Anyone?
Yes, I can see.
Okay. Let's see if I can figure out. I never know. Okay, so maybe it's this thing right here. Spotlight. Okay. Deny, it won't work. Okay, so this is the gene gun. Essentially what happens is we'll have our culture material in our Petri plates. We will actually put that in the bottom of the gene gun. Your DNA, we actually mix it with tungsten, which actually gives it weight. We will put that into a little Swinney filter up on top. And then when we apply a vacuum to a certain point, we'll then actually hit a button. We actually will drive this through by using helium. And that shoots your DNA into the tissue. This next picture down here is actually one of the wheat embryos that you can see all these green foci. These are actually individual cells that were genetically engineered in this situation with the green fluorescent protein. It just shows you the amount of cells that are actually transformed with this material. When we go through selection, not all these will actually continue to grow, only a few of them will, but we will get proliferation. We will put this on 5 to 10 milligrams per liter glufosinate, which will be our selectable marker. Then our culture will continue to grow. They'll start developing shoots. When we put them in light, they'll start greening up. And again, then we transfer those to tubes to start getting these small shoots to germinate. Okay. This is sort of a little more close up with these plants growing. This is what the roots look like. What we do is we look at the roots. That actually gives us an idea as to how well they're growing with the glufosinate. If they are negative, like this one over here, the roots really don't grow that well. If they are positive, the shoots, the roots will actually grow into that media really good. We'll have bifurcations. That's a good thing. And then also on this one, you can see little root hairs. That actually also gives an indication that that root is really healthy in that selectable marker. So that's one of the things we actually look at. For this, then what we do is we transplant these to soil, and eventually we'll get a plant to test. This is a little old, but what we'll do first is we'll paint those with Liberty. This is the active ingredient is glufosinate in this herbicide. The leaf on the top is resistant to the Liberty, so that's a transgenic down here. It's the leaf is a little more yellow. That's not a resistant plant, and we would cut that shoot out. But so we'll go through there first to select for the transgenics. This is the sort of preliminary screening. And then later on, what we'll do is we'll go through and we'll isolate DNA in those. Use your primers to amplify your gene of interest, or the and also the bar gene. So the bar gene, we have set up primers that will actually amplify this. With our co-transformation process, we are using PHC 20, which actually has the maize ubiquitin promoter driving the bar gene. And this gives us a good expression throughout the plant. So we will look for this. If that's positive, we will then go and look at your gene of interest. The gene that I show here is actually an RNAi gene of interest for a virus, but that's just to show you the term what we'll do. We can also use your gene of interest, and we would actually ask you to actually amplify or give us the primer or give us the primer sequence for that material. Okay. So that's one of the things we do. For the process, what we have is this is sort of the timeline as to when you think about the transformations, how long it goes. So initially, before we do the transformation, we have to grow these plants to acquire the immature embryos. That takes between 60 to 120 days, depending on what cultivar it is. If it is a spring wheat, it is really early, but if it's a winter wheat, of course, you got to take into account the vernalization of that material. So we usually plant 5 pots or so per experiment, 3 or 4 seeds for a pot. That will give us enough embryos to actually go through and do a good experiment with these. Day 0 is actually the day that we start with initiating the immature embryos. We'll put those on plates. We'll let those grow for about 2 to 7 days. And then here is Hongju in the middle. She is my lab manager. She'll go through and she'll select for the proper tissue that has good embryo production. So she'll do this. And then we will also bombard those about a week later. And then we start to recover those where we'll take them on a series of different medias. CM4 is our callus induction media. And then after about a week or so, then we'll transfer those over to glufosinate, transfer those over for about 6 weeks, and then we move those to shoot production media. And then after that, once we start seeing some good shoots coming out, then we actually transfer that to another media called shoot elongation media. And so the shoots will start elongating. After this, this takes kind of a long period. It doesn't all go at the same time. So it's either between 8 to 16 weeks. That's about the time we actually select shoots from this material. And then we will transfer those to soil and then to a larger pot. And then by week 13 or 14, that's where we start painting the leaves, actually looking to see whether or not we have transgenics. Okay. And then by the end, we will harvest the T1 seeds for the group. Okay, so that's sort of the underlying area of how we do this. The total time from bombardment is 5 to 7 months for spring wheat, 6 to 9 months for winter wheat. So it's not a trivial process. It takes quite a while. It takes a lot of time for folks to do this. And this is why the SCAB initiative has decided to try to fund these transformation centers. All this takes a lot of expertise. You got to know what you're doing to create these transgenics. And then we also again, with the expertise in our lab, that frees up you to actually do your experiments in your lab. Okay. In addition to that, you also, for us to get the seeds to your laboratory, this is for other people not at K-State, but outside of K-State, we need an interstate movement permit for this. All genetically modified wheat has to have these permits from USDA. And we also treat the gene edited plants as genetically modified as well. Okay. And the reason for that is our transformation process. We have our selectable marker in there as well, but it's better to just treat these all as genetically modified. So you have to go to USDA for this. You or your PI has to be have level 2 clearance for the e-file for APHIS. But essentially, what you do is you have an authentication that you sign into, and then you will actually then go through and go to the right area. It's kind of a headache going through the USDA files for the first time, but I could walk you guys through that if you need it. But essentially, you'll just sign up, write down basically what is in your constructs, what gene you're putting in there, what species you're working with. Of course, we're working with wheat. You will submit that, and then probably about, I think it's about 6 weeks or so, you will actually get verification back that you have approval, and you'll get your number. Once we get that number from you, then we can actually ship the seed out. So usually with this, when we're at about this stage, week 14 or so, we will actually say, hey, we're starting to get transgenic plants back. You should be filing for your e-permit at that point. That way you're not held up. The e-permit is only good for a year. So it doesn't make sense to go for that e-permit when you initiate the project. You really want to get it to where we start getting the seeds back out of the plant. Okay, so that's how we want to do that. Output for our facility. This is just taking a snapshot of the last 2 years of our funding cycle. So with this, we had 3 different clients from the SCAB initiative, and they gave us 29 different plasmids. So that's quite a few plasmids that we're shooting. We've put those into different varieties. This is Bob White Fielder, RBO 7, Forefront, Rolog, Chinese Spring. These are all spring varieties, but we have some capabilities of putting it into winter varieties if people want. With these, we had over 500 separate experiments with over 2,700 bar positive plants putting out. So that's a fairly sizable amount of material that we've actually put out for the SCAB initiative. Okay. And so moving forward, what we're doing is we're also always continuing to improve our elite cultivar transformations. Not all cultivars are transformed competent. A lot of it has to do with how they respond in tissue culture. With the advent of some of these other morphological genes such as GRF and GIF and Baby Boom and Wushel, we can improve that. We've actually shown that we can put that into some of the elite cultivars using some of these genes. We've also looked into agrobacterial transformation. Really, we focused on the gene gun because it is just so good for us to do that. We're very used to it. Agrobacterial transformation, that has its own issues. However, we are converting some of our material to agro transformation if people want to do that as well. Okay. And then we're also trying to enhance gene editing techniques. Along with the gene editing techniques, I do have to say that the efficiency is a little bit less, or actually quite a bit less than putting out transgenics. So if you want to do gene editing, it's more involved. And we have to do many more experiments to get the same amount of plants out. With these, what we typically do is we will send DNA to the provider, and then that way they can actually pre-screen those plants to try to see whether or not they had the edits in there. That may help with trying to with sending the seeds back to the PI. Okay, so like to sort of end it there. I know it's a short talk, but our facility right now, we have Hungju Lee is our lab manager. She does pretty much all the work on this. Although I do have some students, Giovanna Moira, Louisa Adami, and Michelle Wallish. They help out from time to time. They have their own projects in the laboratory, but they also do help out with some of the work that we have done for the facility. And then we have some past members here. So with that, I'd like to stop my slide presentation and take some questions if you guys have any. Let's see, how do I get through all this? Stop to share. Okay. So do we have any questions for anyone? Lawrence, go ahead. I know you have a question. You always have questions.
Okay, my question will be like, you made mention we have Bob White and some other lines that you use for the transformation or for the transgenics. Are there any like reasons why all those lines are better at transformation compared to other lines?
It really has to do with how they respond in tissue culture. So what we'll do is we've done a number of screens where we've taken lots of different varieties and grow them up and then played out the embryos and see which ones respond with the embryogenic response. So if we can get that initiated, that's great. Some of them, especially a lot of, notice some Oklahoma lines we actually evaluated for winter varieties. They just don't respond well in tissue culture. We may get 2 or 3 embryos, whereas embryogenic clumps, whereas Bob White and Fielder will have maybe 50 to 70% coverage of embryos being grown on those embryos. So it really varies.
All right, thank you.
Okay.
Hi, Dr. Trick. It's Bhavit here. I have one question.
Yes.
Am I audible? My voice is little because of I have a bad virus. Uh, I have a question about the cultivars. Is there anything your facility has done with the tetraploid wheat like Chronos? Like, can we transform Chronos or do CRISPR editing Chronos?
I think Chronos could be. I think we worked with that a little bit. The response is probably 10 to maybe 20% of Bob White. So just, so you're going to have to put in either 5 or 10 times the amount of work to try to get constructs back out of Cronus.
Okay.
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 So it really, each one. And that's the other thing is, if you guys have a variety that you guys want to test, we could take some of that seed, grow it up, and then actually perform just a simple test in the lab to try to figure out whether or not they're going to respond in tissue culture. And that usually helps. A lot of times what I do is for a number of these, for especially in your lab, you guys like Chinese spring, right?
Yes.
And that's also another very difficult one to transform. We just need to put a lot more effort into that. But we also suggest, if you want it in Chinese spring, if you can put it into a different variety, say Fielder or RB07, we can put it in there, and then that's sort of a backup. So if something happens with the Chinese spring, we just don't get a lot of transgenics. At least you can have some of these in these other backgrounds that you may not like. You've got your transgenics, and then it would be up to the breeders to actually just shift it into the variety that you want.
Okay. Thank you.
Okay.
Hey, Harold, I got a question for you.
Yes, Paul.
It's a little off topic, but related. I'm not up to date on the stability of transformation these days and gene silencing. What do you see from some of the projects you've worked with on how many generations you can cross and what kind of silencing effects you run into?
We have definitely seen silencing. So you may have have the transgenics working really great in the first and second generation. And then as you get into the third and fourth generation, they actually kind of silence and go away. It's a mix. We also have other ones that we've actually had no problems with continual expression. So I don't know really what the rhyme or reason is of it. It may have to do with some of the backbone material that's actually being put into the plant. Maybe the wheat actually sees that bacterial DNA as highly CG rich and maybe methylates it and actually then turns it off. That's just a guess. I haven't really gone through and looked at that.
Do you see less silencing with just simple edits? I know you'll still have some of your selectable markers in there too, but are
If you're
those any
doing,
better?
yeah, if you're doing gene edits, at our point, we haven't seen any reversions back from those edits back to something else. So if you actually are able to get the gene edits to work, I don't think there's going to be a problem with that. So with gene editing, what you'll have is if you do CRISPR-Cas9, you've got your Cas9 gene that you introduce in, you've got your guide RNAs. They're actually being transgenic. So you'll be putting those someplace in the genome. The first generation, they act, or during that transformation part, they will actually produce the gene edits for you. And then you can breed the Cas9 out of that because the Cas9 usually goes into a different chromosome than what you're trying to target. At that stage, you don't care whether or not those things are silenced. You just get those, you just breed them out of there.
Thanks.
Okay. Emily, do you have any burning questions?
I'm curious, just a comparison of particle bombardment versus agrobacterium transformation. And I know you said you use gene gun primarily. So I was just wondering, what are the pros and cons to both? Because I've heard both sides, but I just wanted to see what you think.
Yeah. Well, the pros of the gene gun, it's fast. You don't have to worry about the biological system too much. It's a physical process. You put it in there. One of the cons is if you want to actually commercialize it, you will have extraneous DNA in there, and you've got to contend with that a little bit. You may have to sequence and find out where that DNA is located. That is one of the problems that we have. Realistically, most of the stuff that you're working with these projects, they're still in the developmental stage. And so if a company wants to come in and actually use your gene, they will also do it in their own system. They'll remake the whole transgenics because they want to make sure that their IPs are solid. Okay, so that's one of the things you have to worry about. Agrobacterium, a lot of people claim that they're simple integrations. Well, when we start playing around with all these genes to enhance agrotransformation, you're also enhancing the copy numbers that are going in. And so sometimes that can be a little daunting. People think you may get single integrations, but you actually get multiple ones going in. But for the most part, it's assumed that agro gives you much less integrations than the gene gun. Does that help?
Yes, thank you very much.
Okay.
Dr. Trick, I have another question. So in terms of FHB resistance, are there any success stories or transgenic lines that have like really solid FHB resistance that can be crossed to like adapted varieties?
As far as I know, things look really good. Now, again, this is where we come with a confidentiality where I can't really
Yeah.
discuss other people's work, but we have seen some of the transgenics that we produced look really good in the greenhouse. But when you take them to the field, things fall apart. So, I still think that we're looking. We still need something out there that's really good. Some of the transgenics have promise. I think actually probably combining some of these transgenics together might be a good thing. So if we can get the some of the different PIs together that have these different genes and actually put them together, that may be a beneficial effect. But yeah, we have some genes that look promising, but nothing like Roundup Ready genes or anything like that. Yeah, no silver bullet yet.
Thank you.
Okay. Any other questions?
I have one small question related to genotyping these transformation editing events.
Okay.
What's the according to you, what's the best generation to we should genotype to achieve a homozygous, like a homozygous edit?
Uh, that I'm not really sure. Yeah, the edits that you put in should be they'll definitely be fixed. So I would probably say the second generation, if not the third generation, you'll be able to see some good material there. The question is what edits you have. Not on the fusarium side, but on some of the other sides that we've seen for virus resistance, we've actually had edits in all 3 genomes, which were positive. Other edits have been single or on,
Okay.
or maybe just at 1 or 2 of the genomes. But you know, basically those edits, you just treat those like another genotype, and so
Okay.
that's how that's how I'd work those.
Awesome. Thank you.
Okay. Anything else? Amber, if we don't have anybody else, do we go ahead and just cut this off?
Yeah, if there's no more questions, yeah.
All right. Okay. Well, thank you all. I know this is probably a pretty boring presentation, but I just wanted to show you what we were doing in the laboratory for the SCAB community. And hopefully take away from this is that it is a very detailed process of how to do the transgenics. But we're here for you guys. So think about your projects. If you do have some genes you want to put in, please give us a call, contact us, and we'll see what we can do about trying to work those out. I guess one last thing I want to say is with your genes, it's really important because you see how long this process takes, 6 to 9 months. It's really important for you guys to go ahead and sequence your plasmids, make sure they're in the right orientation, make sure that they actually are correct in the when you put it together. So the promoters and terminators and the gene itself. We had several early on where we actually generated plants for everybody. And then 6, 7 months down the road, we have seeds ready. And they say, oh, never mind about those. We may have had a mistake. Here's the right plasmid. And so we had to do all these things over again. So it's important to really get your constructs right before you send them to anybody, whether it's us or if you're doing it by yourself in a different system. Okay. All right. So I guess we can go ahead and stop then. And you guys have a great rest of your week, I guess.